Hello and welcome to Nikolai's genetics lessons, and in this video I'm going to talk about unusual phenotypic ratios 9 to 4 to 3, and example would be if we would cross this horse that is dark brown with another horse that is also dark brown in the following generation, we can get following phenotypes: 9 that is going to be dark brown, to 3 that is going to be tan, and 4 that is going to be white. So those numbers here, 9, 3 to 4, we put them in the order how numbers are reduced. So basically, if you watch my previous videos about unusual phenotypic ratios, you have a good idea how to solve this problem, and I recommend you to try to solve this problem on your own first. So those people who didn't see my previous videos were about also unusual phenotypic ratios, and the most famous ratio of the hybrid cross you know as 9 to 3 to 3 to 1. This is classical example of the result that Mendel got crossing a pea with different color of the peas and different shapes. So he had two traits, and this is what he got. If you may think that here we have three different alleles that produce these colors. For example, if you think that here, for example, allele A is dominant over the allele B, and allele B is dominant over the allele C, this is not so, because as I said in my previous videos, I already show different phenotypic ratios such as 15 to 1 or 13 to 3 or 12 to 3 to 1, and also we had such a phenotypic ratio as 1 to 4 to 6 to 4 and to 1, and 9 to 7. So do you find anything in common between all these ratios? These ratios are so different, so it seems that there is nothing common among them. But if we add all these numbers, for example, here we are going to get 16. We are going to get 16 here, we are going to get 16 here, we are going to get 16 here, here, and here. And today's ratio falls under the same category, so 9 plus 4 plus 3 would be 16. And those people who watch my previous videos know that when we got 16, most likely our trait under control of the two genes. So this was my first hint, and the second hint would be here, instead of 3 colors, and you may think that each color would be produced by different gene, we actually have 2 colors and absence of color. If you think that white color is a color by itself, actually it is not. This is absence of any pigment. So when pigment is not present, when pigment is not produced, then we would see, for example, people would be albino, and horses would be white, and many other animals also would be white. So white is not a color, it is absence of color or absence of any pigments. So that's why we have 2 genes, but we have 3 colors. So what do we have here? We have here two genes. Let the first gene would be gene A, and the second gene would be gene B. And we have two alleles for each gene. So gene A represented by two alleles, which is dominant and recessive, and these two alleles in diploid organism can produce three genotypes or three possible combinations, which is going to be capital A, capital A, small a, and small a, small a. And gene B also have two alleles, dominant allele and recessive allele, and also these two alleles can in diploid organisms produce three combinations: dominant, 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 recessive, and recessive, recessive. So homozygous dominant, heterozygous, and homozygous recessive. Now, when we know that this trait under control of two genes, let's think. For example, if one horse would be following genotype capital A, and capital B, and if we cross with the same genotype, as you see in the following generation, F1 generation, only one genotype is possible, which is going to be the same as parental genotype, and no variation we are going to get. If we say genotype of the brown parent, which I used for example when we cross two dark brown parents, for example, would be small a, small b, and we cross with the same genotype, which is going to be small a, and small b. Once again, in the following generation, we are not going to get any variation. So if no variation in genotype, that means that we are not going to see any phenotypical variation too. So this gives us information that most likely genotype of the brown parent that I used as example would be capital A, small a, and capital B, small b. And if we cross with the same genotype, so with another dark brown parent, we expect to see following ratios. As you remember, 9 to 4 to 3. So how it is possible? Why not classical 9 to 3 to 3 to 1? And this is possible if we consider that dominant allele A would produce dark pigment, so this allele would produce dark pigment, and recessive allele A, this is going to be the same gene, but maybe slightly modified. Maybe here we would see some mutations, and this protein wouldn't be able to produce pigment in the same quantity, or pigment that is going to be dark, and we would perceive it as light brown. So this allele would produce light brown color. And if we consider simple dominance, here we would see that this genotype would produce dark brown phenotype, and this genotype here, which is heterozygous, also would produce dark brown phenotype. So simple Mendelian genetics, simple dominance. And only we would see two recessive alleles, phenotype would be different, and we would see that color of the horse would be light brown. And as for the alleles B, this can be enzyme that have to convert this dominant or recessive allele in its final form, and if we would see genotype that is capital B or capital B small b, both these genotypes would produce normally functioning enzyme. So this enzyme would convert these proteins in its final form of active pigment. But if we would see that, once again, consider that dominant allele B would produce normal globular protein and recessive allele B would be defective and wouldn't be able to produce normal protein or enzyme, so it's also wouldn't be able to perform normal function of converting this premature protein into the active pigment. So if both alleles would be normal, of course such enzymes would work normally. If one would be normal and another would be defective, still at least we would see that some of the enzymes would be active and would convert these proteins into their final form, and they would express the color. So but if both alleles would be defective, this enzyme would be able to affect these pigments, and we wouldn't be able to see any pigmentation in horses. So we wouldn't be able to see any color at all. So now let's consider, once again, let's return to our cross. So parent 1 here, parent 2 here. As you see, both parents has the same genotype, so parent 1 may produce following gametes. It can be say capital A and capital B. It also can produce capital A and small b, capital A and small b gametes. Also can produce small a and capital B, small a, capital B gametes, and the last variant would be small a and small b, small a, small b. And the second parent, which has the same genotype, also would produce the same type of gametes, because genotype is the same as parent 1. So we may say that this side would be male side and this side would be female side or female gametes, and gametes are haploid. So if we see that here 4 alleles, in gametes we would see only 2 alleles, because gametes are haploid. So once again, female gametes can be capital A, capital B, capital A, small b, small a, capital B, and small a, small b. Now when we build a Punnett square with 4 columns and 4 rows, we are going to get 16 cells. Do you remember my first hint? 16 means that probably this is dihybrid cross. And now we can put genotypes inside these cells. So here we would have capital A, and capital B, capital B. So if here we see gametes, and male gametes are sperm, and here on the side we see female gametes, which is egg cells. Inside the cells here we would see 4 alleles. That means here we are looking for the genotype of the organism that is result of fertilized egg by sperm. So different types of sperm can fertilize different types of egg cells. So here we would have capital A, and capital B, small b, capital A, small a here, and capital B, capital B, capital A, small a, and capital B, small b. I will speed up a video to save a time. So I finished table, and in the first cell, what phenotype we can see? We can see that here we would see two dominant alleles which produce dark brown color, and two normal alleles which produce normal enzyme which is needed in order for the color to be present. You may also consider these two alleles as a switch on and off. Homozygous dominant means on position of the switch, and homozygous recessive means position would be off. Switch would be off, so no matter what these two alleles produce pigments wouldn't be produced without enzyme present. So here we would have dark brown phenotype here, dark brown phenotype here, and here, dark brown phenotype here. But let's take a look what phenotype we have here. This genotype is able to produce dark pigments, but this genotype doesn't have functional enzyme, so we wouldn't be able to see any pigment here at all. So the color of the horse would be white, and dark brown again, and here what we would be able to see, one allele that would produce dark brown, one allele that would produce light brown, but no functional enzyme, so no pigment would be produced at all. And dark brown here, dark brown phenotype here. And let's take a look what we have here. We have here recessive allele, another recessive allele, that means light brown, and two functional enzymes. So in this case, we can say that color of the horse would be light brown, and light brown here, the same phenotype, dark brown here, and once again here we see that non-functional enzyme, so color of the horse would be white, and light brown here. And in the last cell we see two alleles that can produce light brown pigment, but non-functional enzyme here. So once again, this horse wouldn't be able to produce any pigment at all and would be white. So what we have here? We have, let's count, 1, 2, 3, 4, 5, 6, 7, 8, 9 dark brown, 9 dark brown, 2, 1, 2, 3, 4, 4 horses that is going to be white, 2, 1, 2, 3 horses that is going to be light brown. And this is exactly what we had at the beginning of our problem. We crossed two dark brown horses. Genotypes, of course, can be different in order to produce the same, as you see, dark brown phenotype, but we only can get these results in the next generation if both parents, that would be dark brown horses, would be heterozygous for both genes. Then in the progeny we can find following ratios of phenotypes. So how we can call this relationship between these genes, gene A and gene B? We can say that gene B would be epistatic to gene A, because no matter what genotype is here, whether second gene would be normal, then it would allow to express this phenotype. If both alleles would be defective, this phenotype wouldn't be able to be expressed phenotypically. So this gene B is epistatic to gene A, and gene A would be hypostatic to gene B. So gene B, once again, control expression of the gene A. So this is example of the epistasis. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.